So I hope you all can hear me well, even in the back. It's, of course, a very special pleasure to be back here and to address you today. And yeah, you heard a little about the journey that I have taken, and it's been a very exciting journey, and it continues to be a very, very exciting journey. So let me start with a cite from Brodsky, Joseph Brodsky. A new messiah, should he really emerge, is likely to know a bit more about nuclear physics and microbiology, and about viruses in particular, than we know today. So even a Nobel laureate in literature can understand that there are developments in the field. Sven Gard was mentioned. Sven Gard became professor of bacteriology here at the Karolinska Institute in 1947, and the year after, the House of Parliament gave him a personal chair in virus research, and that is supposed to be one of the first chairs in virology in the world. Sven had a long history as an extremely promising scientist, and he had a training together with T. Svedberg, and particularly Arne Tiselius, and I'll mention that a little later. Clearly, Sven is responsible for the choice that I made in deciding on what profession or direction I should take in medicine. So it was in the autumn of 1958 that I heard Sven Gard lecturing, and he said that viruses are ubiquitära in Swedish, ubiquitous in English, that they're present everywhere. And maybe not one of the most common Swedish words, but it stayed with me, but it took me 50 years to fully understand what this meant. Now, I did get involved after my time at the permanent secretary writing books about Nobel Prizes, and that came about in an interesting way because it was Stan Prusner who approached me and said, I'm curious about the 1954 prize to Enders, Weller, and Robbins, where they had managed to propagate poliovirus in tissue cultures, which was, of course, a dramatic observation. And so we did a review, and because all the archives are in Swedish, except, of course, some of the nominations could be in other languages, could be in English or in French or in German or whatever. It's all in Swedish, so I had to translate everything. But eventually we made a scientific article about this, and it was accepted in a regular scientific journal, which was very encouraging. And I realized that this is a material that could be of interest to the broader audience. So then I did a chapter on the 1951 Nobel Prize on yellow fever, and from there on I reviewed all the prizes given to virologists over the years, and that came to form this first book that was published in 2010. And that stimulated me to progress to do other things, also not only connected to virology. However, in this first book, clearly Wendell Stanley was one of the central figures, because in those days one had managed to crystallize tobacco mosaic virus, and of course it was in fact exhilarating to the press and to the public to say, here is a crystal that is alive. If you take it and you let it interact with cells, it will reproduce itself. But clearly the terms were very vague in those days, and Stanley, I think, didn't really fully appreciate the impact of what he had discovered. And what he did not appreciate, what these two people, two scientists, Frederic, Bawden, and Norman Pirie, showed just in the end of the 1930s, namely that tobacco mosaic virus contained 5% of RNA. That in those days was thought to be just some rather uninteresting condemnation, but as history has told us, this is a very, very important finding. And later on, tobacco mosaic virus has remained a very important object for studies of the details of virus structure. Rosalind Franklin, when she had left the DNA studies at King's College, she studied tobacco mosaic virus. She pioneered that in the first crystallographic study. And Aaron Klug, later on, Nobel laureate, also focused on tobacco mosaic virus. And I'll show a picture of that later. But way back in the 1930s, virology was characterized by some three negatives, namely that it could not be withheld by bacteria that held back, that filters that held back bacteria allow the penetration of viruses. They were apparently quite small, and they could not be observed by conventional light microscopy, and they could not replicate in artificial substrates. It's not that's much of an argument for a discipline to base it on three negatives, but things did change. In the end of the 1920s, the idea was developed to build an electron microscope. And that was done in Germany by Noll and Ernst Ruska. And this development continued. And Ernst Ruska had a brother called Helmut Ruska, who was a physician. And he recruited his physician at the end of the 1930s to let us look at biological materials. And very fortunately, they selected to look at viruses. And for the first time, one could now see virus particles. And published some 5, in fact more papers, but 5 in particular papers that showed that pox viruses, herpes viruses, the T phages. Look at this, the first time I saw that there was a tail on the bacteriophages. And there were different sizes, different forms. One took tobacco mosaic virus, and all by a sudden one realized that one was working with something tangible, something that you could really. I actually, it's so happening in the book I'm writing now, I'll take the opportunity to discuss this story also. And Sven Gard, in fact, was together with Arne Tiselius in Berlin in 1943 doing electron microscope studies. Think about that situation in the middle of the war. But regretfully, that was one of Sven's mistakes. He wanted to show the ultrastructure of poliovirus, and he found some fibrillar structures. Turned out later not to be the virus, but that can be quite understandable. So later on, as many of you recall, in 1960, we had a negative contrast technique with all by a sudden we could see many more details into the back mosaic virus. This remained really a very interesting target in virology. The second book, which was published 2013, has an emphasis, as you can see, on Watson and Crick. And these were the 2 busts that Watson donated to the Nobel Museum when he was here 50 years after his prize. And of course, to be very clever, I took the front of the bust here on the front part of the book, and on the back side of the book you can see their necks, of course. So their brain is in the middle of the book here somewhere. Anyhow, so that was a good part of this story and a very interesting part of the story. However, when I came to this book, I started to work on the Nobel Prize archives that were available at that time. And that was first in 1960. There was a prize to Burnet at Medaware. Well, Sven Gard, who was very influential in the Nobel work through the 50s, through the 60s, is one of those who was given the most addresses to Nobel laureates, 5 of them actually. And he, of course, was very eager to try to get a Nobel Prize to Macfarlane Burnet, because Macfarlane Burnet is really the great philosopher in virology, and he introduced so many fundamental of it. But he also, and here you can see Macfarlane Burnet together with Ernest V. Goodpasture from Vanderbilt University. He is the father of the embryonated hen's eggs, which was very important in the phase of virology in the 1930s and 40s. But Burnet was a fantastic philosopher, and he was also emphasizing you have to look at the virus as an organism. And when you look at processes like disease, you have to look at the balance between the host and the virus and see what's in them for both of them. So essentially a virus that's caused too much of a disease is not a very successful virus. The most clever one is something like common cold virus that could spread and replicate. Always to have an evolutionary perspective was his message. And sometimes, of course, there are like cul-de-sacs. I mean, if you have tick-borne encephalitis virus, that the spread that gives encephalitis in man, that was no interest to the virus, but it's an accident of nature. So, but then in 1960, Sven managed to get Burnet in there, and that is based on one paragraph in a book in 1949 together with Frank Fenner, and it had to do with immunology. And there Burnet postulates that there must be a phase in which one develops tolerance during late phase of embryonic development, because otherwise we do react to our own tissues. It was just a postulate, and that was later shown, of course, by Medaware and his collaborators. And before leaving tobacco mosaic virus, just to mention that these three gentlemen were very close to get a Nobel Prize. They could show in the middle of the 1950s that if you extracted RNA, clean RNA from tobacco mosaic virus, it was infectious and it produced complete particles. That is on the same caliber like Avery's discovery of the DNA from pneumococci, but eventually they never received a prize. So the third book, as was mentioned, all by a sudden it was prizes between 1963 to 66. 63 is a prize in neurobiology, so I have to try to learn a little about how that discipline has developed, and I got some good assistance of people sitting in the audience that could read the text and see if they were correct. And then I wrote about cholesterol, which is a 1964 prize. But 1965, I'm back to my home turf because that was, of course, Lwoff, Jacob, and Monod, molecular biology again. And André Lwoff is one of those other real great giants in the virology, a great philosopher, and he was the first one to give an acceptable definition of a virus, as you say, infectious, potentially pathogenic nucleoprotein entities possessing only one type of nucleic acid. This statement may be up for a challenge in the future, but so far it's managed to survive, which are reproduced from their genetic material and are unable to grow and undergo binary fission and devoid of an energy creating system. This is a great definition of a virus, and it was actually just at the time when I entered the discipline. So presently, I'm working on book number 4, which is 1966, 67, 68. So if you really want to have a challenge when you retire, do something like this where you have a 50-year secrecy, because each year a new occurs, and a new occurs, and a new occurs. So you can keep going until you hit the wall, essentially, right? So anyhow, in 1966, it was a prize to Peyton Rous for the discovery of a tumor-inducing virus. So chicken sarcoma, and the breast map, extracting this and grinding it up, and in the filtrate, you could show that you could transfer the tumor to other birds. To be fair, it wasn't the first time that this was shown, because in 1908, 2 Danish scientists, Altman and Bang, also did filtration experiments, but that was with leukemia, and in those days, leukemia was not considered to be like a cancer. It was something different, and therefore it's not counted. But they were actually first, and Peyton Rous, he refers to Altman and Bang in his article, actually. And the interesting thing about Peyton Rous, because he makes the discovery in 1911, it takes 55 years before he gets the Nobel Prize in 1966. And even at that time, he hadn't fully understood what the impact was, because he didn't accept that the cancer was due to changes in the genetic material. He had some other ideas. However, it was followed up, and by use of Rous virus, Michael Bishop and Harry Varmus managed to show that it was due to that the virus had picked up a cellular oncogene and moved that over to the new target cell, and that was a cellular gene in a new form that was the cause of the cancer, and that was very, very dramatic. It's a good example of a horizontal transfer of properties by use of a virus, a phenomenon that we will come back to repeatedly in the following. I want to give you the background, also emphasize this particular prize, because it is relevant to what we're going to discuss. So Altman and Cech got a Nobel Prize in chemistry in 1989. What did they show? They could show that RNA can both serve as an information molecule, but can also function as an enzyme. And this opened up the possibility to understand how on earth could life really start. Because if you have the same molecule that can both store information and be an enzyme, you can understand that the whole thing can get going. And that's the reason why we talk about the RNA world. Now, Craig Venter, when he had finished the human genome. He wanted to do something different. And so he said, I'm going to make an inventory of all the genes in our water, in the oceans. So he took a sailing boat, Sorcerer 2, around the world, and every 200 nautical miles, this is good sailing, I tell you. This boat makes on 12, 14 knots and takes the wind very well. And on board there is this a series of filters. So we take every 200 nautical miles, we stop the boat, take up 200 liters of water, pump it through all the filters, freeze down all the filters, and then take them all back to the laboratory and study all the genes. Some challenge, right? So, so it so happened that I happened to love sailing, and I thought sometimes maybe I should have chosen another profession, like being a marine biologist or so. But why is that? If viruses are everywhere, you can look at viruses in water. And so I've been the privilege of sailing a number of legs around the world. And what really struck me when we finally made the analysis of what the genes that are there, some 30% of genes that we said, no, they must belong to viruses. An enormous, enormous impact. And then it finally struck me, what did Sven mean by ubiquitous? He means exactly what it says, that wherever there are cells, there are viruses. And therefore, you have to take the tree of life and you just add these huge balloons along all the bacteria, archaea, and eukaryotes, and these are viruses. Millions and billions of viruses. So, and also before progressing, let me really nail this down. And that is that we always talk about evolution as you're fighting for your existence, and the one who is most competitive will have the upper hand. So one can cite Tennyson in his In Memoir and say, nature is red in tooth and claw. But equally important is that it's green in mergers and acquisition. If you can collaborate, and then you can also survive. And it's just as important as competition, but it's very often forgotten. So the interdependence of life form is more the rule than the exception. And this is important when it comes to viruses. So let me nail this concept really. We talk about vertical gene transfer from parents to offspring to further offspring and so forth. But what we don't emphasize is the horizontal gene transfer. And this was really emphasized by Lwoff when he discovered lysogeny and could show that the genes could stay behind in a dormant way in the bacteria and could spread over to other bacteria when activated. So horizontal gene transfer is probably even more important than the other vertical gene transfer. And also to just recall your memory, we have examples where with what we call endosymbiosis, mitochondria was originally a microorganism that came in, became a part of the cellular cytoplasm, and became a bona fide member of the cell, an important member of the cell, an energy generator. And one third thing that I love to emphasize on that is that all cells derive from cells. So all cells in your bodies, they all go all the way back to the first cell that was created 3.8 billion years ago. Isn't it mind-boggling? That's truly the original sin. Now, David Baltimore is one of those also very tall figures in virology. And the reason why I emphasize this is that he made a very, very clever system for see how you could classify it. And you center everything about messenger RNA. And I call this the toolbox of nature because you find all forms of DNA, single-stranded DNA, double-stranded DNA, single-stranded RNA, plus and minus forms, and double-stranded RNA. Everything is here. And there are a number of different enzymes. And sometimes if you're like a vaccinia virus and you want to be able to propagate your DNA in the cytoplasm, then you have to bring along the enzyme that can replicate DNA. If you are like an a retrovirus, of course, as the name says, if you're going to replicate, you first have to copy your RNA into DNA, and you can integrate in the cell DNA, and then the whole process can get going. So there are all these examples of different, very specialized enzymes that are critical. And in essence, this is the various tools that nature can use in furthering evolution in different ways. And we'll come back to some of those later on. Now, since I left virology in 1997, I mean active experimental virology, there are a number of incredible developments. First, the megaviruses. Who would have thought that there are viruses that contain many more genes than what bacteria do? And then there are, of course, the nucleic acid sequencing with incredible consequences, what we call the metaviromics. And the authors that write about it talk about a tectonal shift in understanding of virus evolution. And that is a very strong word, because, you know, in the 1950s, when one finally could discover the magnetism of the Earth, one could show that, yes, Wegener was right. The whole the land masses move around, and they can join and they can part. And today, of course, we know that this joining and parting of the land masses has occurred at least some 4 times. But no one believed this. Wegener, he died in 1940, so he, although he made this prediction, he was did never know that he was proven right later on. So it is truly a tectonal shift. It gives unique insight into the archaeal virus, because wherever there are cells, there are viruses. So we have archaebacteria that can grow under most remarkable conditions. And of course, they have their viruses too. And these viruses can replicate even on those rather bizarre conditions like temperature of over 70 degrees Celsius and so forth. And what about ourselves? The human virome. And I'll exemplify this as we go along. Now, the mimivirus, it was quite dramatic. A huge virus with a lot of fimbria on them, and turned out to be a number of them. And in terms of size, these are what the normal viruses are, in that all by sudden you're the virus that have all these genes. So how do you distinguish a virus from a bacterium? But in this case, it had unique features, so it is a bona fide virus. It's classified as a virus and has its origin in the world of viruses. Now, there is one person, Eugene V. Konin, who is writing intensely about the whole evolution of viruses, and I'll give you 2 references here. One is from Virology in 2015. That is a paper that I have drawn on a lot in this lecture. And then they said just 3 months ago, a theme issue in Virus Research, a special issue that has to do with understanding virus evolution, and they have written the introduction to that. And here you see the word metaviromics, a tectonal shift in understanding of virus evolution, the introduction. So, how many viruses are there in the world? Well, this is just a calculation I did way back when it comes to water. 10 up to 6 microorganisms per milliliter in water, and 10 up to 7 virus particles. So you multiply this with all the water in the world, and you end up with 10 up to 24 virus particles, 10 times all the stars in the universe. You can use all these parameters. You can measure it in terms of how many hundreds of thousands of blue whales. So you can put all the virus particles in a long row and say that will reach the end of the universe. I mean, these are simply big numbers. And potential host, if we try to look at that, so I mean, animal species have 1.5 million, but perhaps we're up to 20 million different insects, 0.3 million plant species, microorganisms. We have no idea how many microorganisms are out there. And half of all organic material out there is either microorganisms and their associated viruses. So how can we talk about ecology by looking at one species of a woodpecker or something like that? Ecology is something much more complex than what we usually associated with. So, but in microorganisms, 6,000 species, but identified, but there must be millions and millions of them. And how many viruses per host? Well, just in us humans, we have more than 200 viruses. So you end up with enormous numbers. And here is a recent article one tried to illustrate this, and of course it's not a very meaningful formulation, but here is known classified virus with 0.05%. This is what the International Committee for Taxonomy of Viruses is working on. And that probably, represents this. So by definition, 99.99% of all viruses are not known at present. We're talking very big numbers. And this actually is a bit of a problem for taxonomy. How are you going to handle this? One is to really start to move, how we have to get all this information. And part was previously you had to isolate the virus, by the old Koch's postulate, and you have to define if it was several different serotypes. So in polio, they defined 3 different serotypes. Okay, if we want to make a vaccine against polio, we have to have all the 3 strains there. So it was easy to say that a species is equal to a serotype. But this doesn't work any longer now when you derive everything from studies of the genetic material. And some traditional virologists are a bit upset about this because they're that. But remember, using terms like species, genus, and family is just an ad hoc terminology. It doesn't reflect evolution like it does in other organic material. Now, this is a slide one could probably speak for an hour about this. And I just want to emphasize there are viruses that can parasite on viruses. There are a lot of whatever replicants or replica and transposition signals like viroids. Here's a piece of nucleic acid that can replicate and can survive in nature, doesn't produce any proteins, but it clearly is a part of the whole web. And then you have like small plasmid, you have a large plasmid. Here are how you have the viruses, the analytic that can destroy the cells, but you have temperate viruses that can stay in. You have viruses that stay in the cells. And it's simply very, very complex if you look at as aspect of autonomy, selfishness, cooperativeness, and you can even discuss what the chromosomes represent. They have a certain degree of independence in our cell. So, and I won't elaborate it, but just to emphasize that there are all these different forms of self-replicating units of various kinds that can shuttle in and out of cells, that can shuttle in and out of the genome, and so forth. So if you now start to think about what about evolution in this very early part? So we say, yes, that was the primordial RNA world. Here is an RNA that in some way can replicate itself, and that is the first definition of life. So life, we'll see that definition later, is a system with a chemical system which allows the replication of a particular molecule. But in the beginnings, of course, it is an RNA enzyme that replicates the RNA molecule that contains information. And so you can follow that step. But then it gets more complicated. Then you came to the RNA protein world. So now, in some way, information starts to flow from RNA to the protein. And there's a development of a genetic code. And of course, also the genetic code has an evolutionary background. And I should actually go on to write about that. When I write about the 1968 prize, which is actually was given to Nuremberg, Kurana, and for the genetic code, there is reason to discuss how could a code really develop. And so they start to get more complex. But once the protein world gets involved, and one can have this various enzymes, you have at an early stage an enzyme that can replicate RNA, RNA replicase. This is probably the most important enzyme of all. That is the most ancient enzyme, and if you're going to follow how evolution really takes off, you should follow the way the RNA replicase developed and what base composition it has. And then as we move on, you come to the retro world, because now we want to get from RNA to DNA, And that's another quantum jump in evolution, early evolution. And then you can get, we see the various forms in introns and retrons and so forth. Finally, we come into the DNA world, and then the way we know the world today. And from there on, it's all started to be encapsulated in a cellular membrane. And the reason for this encapsulation and increase the efficacy is, of course, that it allows for the chemical reactions to occur in a limited volume. But all these various steps here are, of course, very, very speculative. But one can at least understand the logics of this as one go from step to step. So we have the two groups of bacteria, and finally you get the eukaryotes. And there's evidence today that the archaea is much more important, but in particular the Taq phylum, in allowing the development of the much, much larger and much, much more complex eukaryotes. And of course, you have accompanying viruses, archaeal and bacterial viruses, very selfish elements, eukaryotic virus self-element, and then you can also go all the way up to the retroid and RNA viruses. I comment on this in a very superficial way, but to illustrate that there is an enormous complexity. So what about the origin of major groups of RNA viruses? So here is the whole gemisch of prokaryotic viruses. Now, maybe some of you recollect that RNA phages are rather uncommon, but there are, in fact, two forms of RNA phages that have been identified. And they represent the origin of this, and I'm not going to go into how this can grow into all the forms of RNA viruses, or can grow into even double-stranded RNA viruses. But using sequencing of genomes, one can now follow each step in this. And the more viruses we study, the more firmer gets the picture. But this is from one of O'Connor's articles. So that is for the RNA viruses. This is from the same article, emphasizing that it's an enormous complexity of reverse transcriptase generated elements. If we look at the human genome, half of that genome are various forms of retroelements. And you immediately ask you the question, why on earth should we carry along 50% of DNA for which we can see no use whatsoever? Is it just a historical fact, or it doesn't matter that it's a large DNA? But then in that, you find a lot of things like the line element or the alu element and so forth. And you can also find retroviridin. In fact, you find quite a lot of retroviridin. Sometimes you can identify all the three critical genes. Both the reverse transcriptase, the polymerase, the envelope gene, and the GAG gene. Do they play any role? Are they just there? There's some speculation that they may play a physiological role in forming the syncytium in the placenta, but for the rest, there's no real evidence for any particular role. I'm writing, of course, now about Peyton Rous, and then also what about, since we have this human endogenous retroviruses, wouldn't it be logical to expect that they could play some role in the cancer in man? There's no evidence for that. So even though oncogenes was discovered by the horizontal transfer by use of a retrovirus, that doesn't seem to be the case in humans. There are other viruses that cause tumors in man, and that is another big story, of course. So here is the very complex root of retroelement and reverse transcribing elements. There's a plethora of them. In maize, for example, there are about 75% of the genome are those such retroelements. So maybe it's no wonder that Barbara McClintock could find jumping genes in that particular species. So the large DNA viruses, we can do the same thing, prokaryotic viruses. And here, of course, the overall majority of all phages are DNA phages. And there are different fundamental basic structures here. And you can follow that and see how you can alter the megavirales, a lot of different of those. And here is Johan Waddell's and my favorite, adenoviridae, with a beautiful icosahedron with the vertex projections. Here is herpes viruses come up from another. It's a bacteriophage with a kind of T-even phages with a tail. Now, the interesting thing is that if you look at the icosahedron in herpes virus, you find something that is different from adenovirus. In adenoviruses, all the 12 vertices are identical, but in herpes viruses, one of them is different. And that is probably can be explained by this origin, and therefore the vertex that is different is probably the one where there is a motor that can pull in all the DNA to make a head full of DNA. And that machinery has been illustrated beautifully in bacteriophages, fantastic molecular machines. And we're talking about giant molecules that are going to be pulled in there, but nature has solved that. So one can now, as this field grows, you can start to see a relationship between all the different viruses, so their evolutionary origin. And here, when it comes to archivira, we have just scratched the surface. These are just some examples of what they look like. Most bizarre, the ampules, or they are like drops, or elongated. And almost nothing is known about the nucleic acid of these viruses. It's a field that one must scratch the surface, and the more you want to look at things, the more you'll find unique viruses. And nature is, in fact, very imaginative, I would say. And added to this, when we deduce the protein structures from the genes that we sequence in viruses, 80% of them have no homology anywhere. And we have sequenced many proteins now, and all are through the biological kingdom. So we call this the virus dark matter. But at the same time, one has shown that you may have a preserved structural homogeneity. You can achieve the same function of a structure even though it has no genetic homology. So the possibilities for variation are enormous. Now, Carl Linnaeus, the great Swede, who classified everything, and animals and plants, but what about minerals? Isn't that the most inorganic thing you could think about? No, of course, there are no indications that biological materia, viruses, can influence also mineralization. So I'm teasing the young virologists out there, why don't you become a geovirologist? And you can find out how all the different kinds of minerals develop because there's a virus present. So maybe Linnaeus was way ahead of his time in a way. Anyhow, we need this definition. So what is definition of life? We have had this discussion for decades, or more for a century. Are viruses live or dead material? And it is, of course, a fruitless discussion, because in astrobiology, say, what is definition of life? It's a replicating chemical system, and I love this, with the capacity to make mistakes. Isn't that a great definition? Everything in evolution is based on mistakes. And the mistakes opened up for new structures, new possibilities, competitive advantages, and so forth. Oops. And already Dobzhansky said it in the 1930s. Nothing in biology makes sense except in the light of evolution. So we have to put everything in a perspective of evolution. And therefore, in the first book, I use this transmutation of Descartes statement. I exist, therefore I think, therefore I exist. So cogito ergo sum, that when it comes to viruses, it becomes existo ergo pas evolutionis sum. Yes, of course, virus is part of evolution. And in fact, in my world of conception, it's probably the most important part in all of evolution. Now, so I'm heavily involved with Craig Venter's operation. I happen to be the vice chairman of his board of trustees since many years back. And after we have circled around the world, I said, why don't you come to Sweden? Because we have a very interesting water in the Baltic. We have a salt gradient, and it's probably worthwhile looking at all the life forms that there are in the Baltic. So he readily fell for that temptation. He happened to love Sweden. So, and that gave me an opportunity to sail across the Atlantic, which, of course, has been a boyhood dream coming through, starting at the Bahamas and all the way over. And then we took the boat here and took it around. And during this time, I took the boat 3 days through the archipelago of Stockholm. And it was absolutely the most magnificent weather ever. And we started out by going up through Björkskär into Stora Nassa, and you know, it's a big boat, 100 feet. I mean, you have to really make care, and it takes down 3 meters down to it. And then we sailed out from Svenska Högarna, I went up to the Frederic, and then I managed to dock it into the very open, inside Utfredel. It's one of the most magnificent archipelagos we have. And the weather was like this. We took the kayaks, of course, and kayaking around. And then Craig said to me, because we're going to next out to our summer house on Bleeders, and I'm going to take a kayak. I'm going to kayak to your home. Okay, you can do that. You'll come about 1/3 and we'll pick you up. And off he went with some of the crew in the kayaks, and then we picked them up 1/3 away. And then Craig said to me, I think I found the essence of life. Marvelous. Tell me, what is the essence of life? The essence of life is a naked skin against a smooth rock. Think about it. To me, it stays with me. This is an exciting reflection on the meeting between the hard and the soft. And so anyhow, we also took the boat, and this happened to coincide with the Volvo Ocean Rates. And so the king was on board, and I had found out which was his favorite whiskey, so we had a very good time actually on board. Now, human body, the final part of this. Our body composed of 10 to 100 trillion cells. We have 10 times more microorganisms. That is more than 100 trillions. And we have indication of 500 to 2,000 species in the gut, separately on the skin, etc. And these are microorganisms. But how many viruses are there in there? And we just started to make the virome right now. And there are, of course, all the different phages that connect to all the different microbes that we have. And, of course, they always do it together, the viruses and the microorganisms. So whatever we do in studying the microbiomes, we have to also include the viruses, you know. And so we know that this has a lot of beneficial effect in healing our wounds, digesting our food, lots of other things. And just by way of example, so Craig is an incredible entrepreneur. And not only has he managed to have this independent institution of the G. Craig Vance Institute, he also started something called the Synthetic Genomics SGI Institute. And more recently, about 4 years ago, he started Human Longevity Inc. And who doesn't want to have a long life? I mean, it's a very good and selling title. But what they do is that they use all the modern tools to analyze in the human body. So all the non-invasive techniques. Of course, the complete genome, the whole microbiome and everything. And then 2 years ago, Craig said to me, when we had a board meeting now, you should go through our program. Okay, 7-8 hours, everything included. And you get a thick book like this, and that's your life, or your health. In fact, on the same occasion, Ham Smith, you know, a very close collaborator of Craig's, also taking this, and one found some changes in his lung tissue. Turned out to be quite serious. And this was just accidentally, and one operated on that, and Ham Smith is still there in the lab now, 2 years later, doing his work. He's probably 86 or so now. So, I mean, it's not for free, this kind of screening, but anyhow, it can make a difference. And in the future, what we call precision medicine, we will use the genome for various both prospective and retrospective analysis. Anyhow, in these studies, one also looked at all the different viruses. So it was 8,240 individuals. One found 94 different viruses, 19 human DNA viruses, provirus and RNA virus, in 42% of participants. So yes, half of you sitting here, you have viruses in your body. Of course, we know that we have, like varicella virus, because and hopefully it will not develop the zoster. All the different viruses, but then you come to the end of this, but influenza virus, how on earth? This is a DNA analysis that cannot be. Turns out that this individual had been immunized with an experimental DNA influenza vaccine. And that can be detected many years afterwards. Think about the sensitivity of the technique. I mean, it's unbelievable. So that's, of course, in the future when we're going to test our blood donors, we'll have to do the genome and then look at that and see, make sure that they don't have an hepatitis B, hepatitis C virus or anything like that. So here comes the question. Of course, there will come a time when I would like to, first of all, try to understand a little of the story I'm telling you, and really to take this down in a written synthesis of some form. And I could theoretically do that in book number 4, that I'm a little halfway through that book right now. And as I mentioned, I'm going to study, now write about the genetic code, and it could actually fit in there. But if I wait to number 5, however, you never know at this mature age how much time you have. So in 1969, there is a Nobel Prize for the phage work to Delbrück, Hershey, and Luria. And there it would fit perfectly because it all started with bacterial viruses. So probably play a gamble on that and hope that I can make that synthesis in book number 5. But the first thing is to bring number 4 to harbor. So let me finish with another quote of Brodsky, and I happen to like his way of formulating things. The point, however, is that the democratic principle, so welcome in almost all spheres of human endeavors, has no application in at least 2 of them, in art and in science. In these 2 spheres, the application of the democratic principles results in equating masterpiece with garbage and discovery with ignorance. So thank you all for your interest.